So today we will be starting with part C, question number 71. Equal volumes of pH 4 and pH 10 solution are mixed. What will be the appropriate pH of the final solution? Okay, this is a cute question, but there must be a formula to solve this. And as always, I do not know, but we'll try it. So 4 and 10, so we'll come here 5 and it will be 9 and it will come 6 and it will come 8 and they both will meet at 7, so I say it is 7. Okay, I have no other idea. Okay, I don't know. The inborn error of amino acid metabolism, alkapto alkaptonuria, is due to the lack of one of the following enzymes: alpha keto dicarboxylase, p hydroxyphenol pyruvate dihydroxylase, homogenized oxidase, fumaryl acetoacetate hydrolase. So I don't know. It's alkaptonuria. I'll go for alpha keto acid deoxycarbonase because I am guessing. 73rd question. The structure of the protein with 100 residues was determined by X-ray analysis at atomic resolution and NMR spectroscopy. The following observations are possible. A. Dihedral angles. So this is about X-ray and NMR spectroscopy. I have not studied about this. We'll study it later. I am doing the book about techniques, right? So it will come in it later. Right now I have no idea. So I'll just mark it I don't know and move on, because that's what I do. Different arms in the tRNA structure are shown in the column A, and specific signature in the different arm is in the column B. So what do we say? I don't know. Since some coenzymes that serve as transient carriers of specific chemical groups are shown below. Coenzyme A, flavine, FAD, pyridoxal sulfate, NAD. So electrons, chemical group transferred electrons, acyl group, hydride ions, amine group. So these are both adenine dinucleotide. Okay, this one has phosphate and this is coenzyme A. I say coenzyme A with 1. There is only one option with coenzyme A with 1. I have no idea, but I am doing it because I am just like that, and this is not going to mark a big role in my future, but I will know where I stand. So in 2 years I'll have some progress to show. Okay, thermodynamics of the protein folding is depicted as free energy funnel below. Given below is the region of the diagram. This is the region of the diagram. Percentage of protein in native concentration, energy. What kind of weird ass diagram is this? So X, Y, native structure. X column represents, and what Y column represents. So A represents what? Structure of highest entropy, nahi. I say D suggests that. Okay. And what is C? Discrete. What are the options? Molten globule. I say it is B. Let us check out the options. So, anyone has D as 2? No. So my option is wrong. D is 2 is not correct. And B is 3. B is 3. One of them has B is 3. So I'll go for 1. 76th one. 77. Table below show the list of organelles, column A, and signals, column B, that target protein and of the organelles. Lysosome, mitochondria, nucleus, peroxisome. Stretch of amino acid sequence rich in LYS and ARG residue. C-terminal tripeptide, N-terminal amphipathic helix rich in LYS and ARG. Mannose 6-phosphate. What a question. What a question. That's what I'm saying. What a question. I don't know the answer. Following statement are made the chromatin remodeling. I'm like, why am I even doing this? I don't know anything. Remodeling. Combination has all correct answers. So local chromatin. So what I can see is B, A. Okay. Let's see which one of them is incorrect. Okay. Several histone variant exist which replace the standard histone specific. Nahi, this never happened. Chromatin remodeling is, I have no idea about chromatin remodeling. Okay, but as I have crossed out D, I'll mark second one because let God have some fear instill in me when I see my marks in negative, and then I will have some fear in my life to start studying. In an experiment, the student has infected mammalian host cell with cytoplasmic RNA virus. The virus was monitored by measuring the intracellular viral RNA at different time intervals. It was observed that the RNA what is the RNA progressively went down with time, particularly 12 hours post-infection. Following are the possibilities which can explain the observation. The virus infection triggered the upregulation of miRNAs that might have downgraded. Virus might have might encode miRNA. One of the viral protein inhibit replication of the viral RNA to restrict rapid. I don't know. Next. Irrespective of the chromosomal configuration, a single X chromosome remain active. Okay, in all diploid humans, somatic cell lines. Which one of the following mechanism best account for? Maternally inherited X chromosome is developmentally programmed to remain. Best accounts, meaning which one is true. Developmentally programmed to remain active by avoiding DNA methylation. Chromosome specific expression and binding of ROX1 to one of the X chromosome protected from cyst mediated silencing. I need to study about this. The T6 gene produced just enough of the cyst antisense RNA to block 1,6 locus. A cell produced just enough of the blocking factor to produce. I'll go for this because this is the only one that did not complicated matters. In eukaryotic cells, replication initiation from a replication origin occurs only once per cell cycle, and S phase CDKs play a vital role in regulation of DNA replication in budding yeast. In budding yeast, a protein complex known as the origin of ORC is regulated. Replication does not start in mRNA because I don't know. What kind of questions are these? Measurements of the rate of actin treadmilling in vivo show that can be several times higher than that can be achieved in the actin vitro. Wow. It must be something. The E. coli, in a strain of E. coli, a fusion between the LAC and TRP operon took place, and a new locus structure is shown below. The single strain LAC wild type TRP operon. Okay, good for it. Little potential scenarios. Tryptophan will be synthesized in a medium. See, I have not studied about this, so I do not know about this. Next question. So let us just guess. Okay, they do not have a wild type. I basically do not know where the wild type was going to be and what the wild type was going to do. So I can only guess here. So tryptophan will be synthesized in a medium containing lactose and tryptophan synthesis will be repressed in the medium containing glucose. Tryptophan synthesis will take place only in the absence of sufficient tryptophan medium. I'll say this because I do not fear death. Following statements have been made about removal of supercoiling produced by DNA unwinding at the replication fork. Okay, topoisomerase 1 and 2 can remove positive supercoiling. Okay, good. So I'll mark this and this. So 1 is not the answer. Supercoiling topoisomerase by tweaking either or both standalone by blah. True. C is the one. C is the one, and 2 is not the answer. So let's look between A and B. Problem of DNA supercoiling is valid only for circular chromosomes. No. Accumulation. So B is not the answer. 2. So C is the answer. Accumulation of supercoils is a result of DNA helicase during this blah blah. Okay, next. Phosphorylation of the alpha subunit of ELF2 at SIR51 position of Saccharomyces cerevisiae leads to the concentration of ELF2B and a guanosine exchange factor. The phenomenon is, who knows? I don't. E. coli mutants isolated from genetic screen showed following class of mutations. A point mutation, I don't know, basically deletion, For E. coli chromosomal DNA replication, which one of the following statement is true? DNA polymerase 1 is the main polymerase required for DNA replication. E. coli. Following observations were made about the variations among genome and of eukaryotic organisms. A single nucleotide polymorphisms are, I don't know. Genomes, I have not studied this. Tandems and repeats sequence can be shown or the polymorphic or mitotic combination. I don't know. I cannot even guess this. There are a number of specific T cell surface molecules involved in various functions of adaptive immune response. Column X represents the list of T cells. Wow. Just wow. I don't know what T cells does. They are asking me about CD28, CD8, CD154. Like, I would know. I say A, this. And it is wrong. So wrong. Now, I say this. 1 is 4. Oh, there are 2 options. So this eliminates this and this option. Let's complicate a little bit. I'll go for this. C2. Anyone C2? Yes. They both have C2. Wow. Will not let me live and you will not let me die. I'll go for D1. This one. Listen, I do not know. I have not even read the options. Okay. I am just taking a wild guess. It's not even guess. I'm just throwing my tick there. In an experiment, it was observed that the protein was upregulated in the cancer tissue that showed correlation with disease progression. Following are the few possibility which can explain the observation. Good for it. The extracellular matrix contain a number of non-collagen. I have no idea. IDK. If you want to read it, just take a screenshot and read it. If you do not want to read it, then we'll be skipping it like me, like I am doing it. Following this, as protein receptor, transferrin receptor, I have never, steroid receptor. I'll just take a world class guess and I'll go for 3. In a laboratory experiment, it was observed that the virus A and virus B could infect a mammalian host cell, which infected individually. Interestingly, if the cells were first infected with virus A with large MOI, virus B failed to infect the same cell. If virus B with large MOI is added first followed by virus B, both viruses can affect the cells. However, infection with virus A was found to be less extent, considering X and Y are the receptors and co-receptors which may be involved in the virus. So, explain the possible observations. Virus A uses X as receptor and V as co-receptor. Virus B uses exclusively Y as receptor for entry. Both virus A and B need X receptors. I'll say this and this. A and B option. Okay. Question number 94. Whilst testing the effect of several potent anti-cancer compounds on cycling human oral cancer cells, a student observed that a major percentage of cells showed dose-dependent cell death after 12 hours of drug treatment. However, the remaining cell repopulated and clustered once the compound were removed and the cells were cultured and incomplete medium. The students made the following assumption. I don't know what is this. I cannot even guess. When 8-cell embryo of blah blah blah. I don't know. Let's read if you want. I don't know. During the wing development of chicken, blah, 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 blah. I never read Dev Bio. And I don't think I ever will. I hate it. Which of the following statements regarding the generation of dorsal ventral axis in Drosophila was made? Ghrelin protein move along the oocyte. What the fuck? Maternal deficiency in the Ghrelin or torpedo gene cause ventrillation embryo. Ghrelin is active only in new cell. Blah 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 blah. Blah. A specialized area of heterogenesis involved in the misregulation of endocrine system. Which of the following statement regarding the endocrine receptor is true? Okay, I don't know about hormones, but I'll go for they do not mimic. Nahi. This is an easy answer. Let's go for they can act as antagonistic and inhibit. 99th question. Given below the statements which related. What the fuck? What is this? Plant physiology. Following demonstrates the function of different genes to become the trophoblast in the inner cell mass blastomere during the early mammalian development. This is morula, the trophoblast and ICM trophoblast. Okay, so YAP this and CDX and EUMers. Okay, but it goes to nano or OCT4, ICM, some from somewhere STAT3 is there, in ESC is there. Okay, so is on the above figure, which of the following assumption is correct? The interplay between CDX and OCT4 can influence the formula of ICM, whereas CDX2 and OCT4 can influence the formation of yes. ICM would form even if expression of OCT4 was inhibited. Yes. YAP and upstream components in CDX can be inhibited by NAGO. No. The expression of STAT3 is optional for maintaining STAT3. Where is STAT3? Optional maintaining pluripotency of yes. We'll tick this. Okay, so that's enough for today. We did till 100th question. We'll meet tomorrow for from 101 to 125. Okay, so goodbye, bye.